Okay. Hello, students that are hanging out in the virtual classroom. I see, are there 12 of you? They're all muted. So, which is good. So one thing I've noticed in my past history of using Zoom, not for teaching, but in meetings, is that if everybody has their mics on, then there's a lot of background. So I set it up so that when you enter the meeting room, you guys are on mute, and there is a chat button. Has anybody used Zoom before?
Yeah.
Oh, you guys have more experience maybe than I do. That's good. Oh, for another, yeah. Well, we'll all learn these things. So the chat button is a way for you guys to talk to me. Yes. I am going to record them. I have it set to record, so hopefully this is a good trial to see if it works. The other option is, I kind of hate to give the lectures twice, but I could record the PowerPoints themselves too, but I want to continue meeting at this time, okay? So let's talk about what's going on first, and then the students that are on site, they're going to take their quiz while the rest of you are just going to hang out and actually maybe answer some of the questions I've posed. So what I want to know from everybody is if the time we are meeting normally for class, right, Monday, Wednesdays at 4 o'clock, is that still good time to have virtual class? Okay. And you guys on chat, which I gotta figure out how to open up. There we go. So far I'm seeing yeses. Excellent. So that's one thing that we don't have to worry about. And like I said, I will record the Zoom classes so that, you know, if you need to go back, you can. One of the nice things about online, and it's, I mean, I tell you guys all the time that you can email me with questions, right? But we can set up even kind of a discussion forum that we can use to talk to each other. There is chat also on Titanium. So I was thinking there's actually two ways. There's chat on Zoom, and I believe you can go into chat groups, okay? But there's also chat on Titanium. So what I want to do, because you guys always get into groups to do our discussions, I want to figure out how to group you so you can talk to each other and, you know, you don't have all these competing voices. And then as a large class, we'll get back together and we'll do like we normally do. Yeah.
Yeah.
So we'll see how, I'll let you know. I mean, we still got to work it out. So, and everybody has access to either a desktop or laptop computer? Yes? Okay. How about online? Does everyone have a desktop or laptop computer and internet? I actually have a student in my other class that told me that she and her husband, they moved recently, and for Lent, they gave up the internet. Yeah. I don't know. I assume she's using her phone, or I don't know. Well, I think that they literally moved and just didn't have the internet, right, put into their home or wherever they're living. Yeah, right. I know. So I don't know what she's going to do. And so my other college, actually, we were supposed to meet this week. Yesterday, they decided no, they canceled classes for this week. And they bumped it up here by a day, I think. So, what else do I have on here? Okay. So, what's what I'm going to do? So in terms of scheduling, so Wednesday will be a discussion virtually with all of you, just like, well, sort of normal, okay? Next Monday, there are no classes, virtual or otherwise, okay? Yeah, they're giving instructors, though frankly, here we are, we're supposed to be teaching virtually this week, but they give us 2 days off next week. I think they had already planned to do it, so anyway. So we are not meeting on Monday. So we will meet on Wednesday, but if you look at the schedule, we are supposed to be having an exam. Now, this is what I was thinking, and I welcome input. I was thinking that we would have the discussion of JA5 Part 1 on Wednesday. We would do JA Part 2 online in a discussion forum, okay? So we wouldn't meet, but we would talk online and share information online. And then Wednesday, we could meet and kind of have a review and like, what the heck is, you know, kind of catch everybody up, because it's, I don't know about you, but you know, I'm juggling technology, I'm juggling 2 colleges and also, you know, other duties I have to that other college. So, you know, I assume that you guys are a little weirded out by this whole situation like I am, right? So I know that your spring break starts at the end of next week. I was thinking of having the exam. There's 2 options actually. One, have the exam, you know, it's going to be online, have it so you can take it that Friday, okay, online, or literally have it so it is open starting on Thursday or Friday of next week, the entire spring break, and you have it done though when we come back in April, whatever the date is, 30, the 4th, 6th, April 6th. Yeah, so you'd have to do the exam too, right, by the time we come back to school. How's that sound? Yeah, that actually gives you more time. And it's going to be timed. You're going to have the same amount of time to do it as you do in class. So in oh, I'm going to cut and paste it into an online test with essay questions. So has anybody, is there, how much, it says you have 40 lines to answer something. Is that enough? I know. I'm just, hopefully, you know. You think so? Okay. Whoever goes first and runs out of space, let me know. Yeah. Oh yeah, I got that. Let me think if I have. I didn't know, probably not, because. Yeah, what'd she do? Because she.
We have an exam next week, right next to the central exam, and she's making us download a PDF once we open up the test, and then we can edit that PDF on an iPad or print it out. She gives us an extra 30 minutes after exam, basically, to get the drawing in and turn it in.
So you're going to end up scanning it or working on it?
Or making a PDF anyway.
Okay. Okay. So you're actually going to download like a Word document, right? So you can actually edit it. Okay. Okay, that's a good idea. I'm still working on the exam, so I had to edit it anyway. So I will, you know, look at the questions and make sure, you know, that's a great option though, to do that. That's smart. Yeah, we could do, that's actually a great idea. I can do a little like review quiz so you can get some review in and also practice, you know, I mean, you know, half of them are practicing already with the quiz today, but that way everybody can make sure that it works for them. Okay, do you have any questions anywhere at home or in the classroom?
the duration of the exam will be the same as in class.
Yes, the duration of the exam will be the same.
And then while handwriting, still.
Yeah, actually, type, well, it depends how fast you type. Typing should be
I type
easier.
so slow.
Juice on top of scan zone.
Yeah, can I just write and then turn it in? I write so much faster.
We'll see. I'll see, but
I agree.
really? You guys should all know how to type.
I have those classes.
I didn't either.
25 words per minute is basic.
What?
25 words per minute is actually mostly. Yeah.
I'm sure, 25 words a minute, okay.
It is slow. That was like the requirement for my job. One of my jobs.
Oh, yeah.
Basic.
I don't know.
Super low so that anybody can fill.
Okay, I'd love to have this discussion, but we actually have a lot of other things to do. The lecture today is pretty long. Okay, classroom people, get out your note card. So the students in the classroom are going to take the quiz for the next 12 minutes or so, and then we will start doing the lecture. And what I'm going to do is pull up the lecture slides, open them, hopefully in Zoom, and have them show up in the classroom. Here's hoping. So I'll let you know when they're done. Going to stop my video so they don't need to see me while I'm standing here. Okay, so is it on? It is. Are you all ready? If they are, I can't.
No.
Maybe it must be, but I thought I turned it off. Here, hang on. Hang on. Maybe I can. Yeah, that's okay. We can live with it. Ready?
Why is the template have on this?
I am. I'm not going to. About 3 more minutes. That should be fun. Okay, 1 more minute. Okay, pass them up. So. Yeah, good talking. Okay. Second guessing, I know sometimes it's not good, too much time. When you guys leave, wash your hands. Okay. So let's get into. Sorry, I got distracted. I emailed the chair of the department about Proctorio, so maybe they'll actually invest in it. You know, obviously they have to pay for it. So, who knows? Proctorio, it's the online proctoring tool. Okay. Okay. So let's try this. What I'm going to do is I am going to figure out how to share my PowerPoint. Okay, wherever that icon is. Oh, there are breakout rooms. Well, that's easy. So there's breakout rooms for Zoom. So we'll figure that out. Now I just have to figure out how, oh, here we go, share screen. I have so much up. Okay, this is, I should be able to do that. Oh, really?
Yeah, so just make sure it works on my end.
Okay. Hang on. Ah. Somebody's still talking and I can't quite. Okay. The problem is, this is another reason I want to do this at home. So that I can see where the chat go. How long ago was that? Okay. So everyone at home, can you see the title of the PowerPoint? Type in yes or no. Looks like we're good to go. Oh, you said when she advances?
Yeah, when you advance. the
Okay.
slides.
Let's see. I got a gun. Can you guys see that I'm on the next slide now? Looks like it. Woohoo! Yes. Okay, here we go. This is gonna be. Let's see. Just give me a sec. I know. Oh, you guys can't see anything yet. Well, that I can fix easily. You're all like, but you have to say stuff to me. I'm looking at all the other. Okay. I'm trying to close this so I don't have to see. Hide self view. Yeah, I don't really need to see me. Can I hide all of them too? No. Okay. So if you notice, on one side is the paper we just talked about, even though it feels like it was a really long time ago now, right? So we talked about MyoD and how one transcription factor, right, can actually change the type of cell it is, and that is called induced transdifferentiation, right? So the cell suddenly becomes a different kind of cell. Well, not suddenly, but because of that transcription factor, right? And we use classic textbook type techniques in there. The paper we're going to talk about on Wednesday, which was published about 20 years after the MyoD one, it actually looks at numerous transcription factors, and you're going to discover that they started off with, I think it was 24 different transcription factors and worked their way back, or maybe it was 27, to they eventually found that there's 4 of them. Okay, and they, by transfecting cells with these transcription factors and expressing them, they found that they could induce pluripotency. So they could take cells and turn them back into embryonic, stem cell like. Okay. The techniques and this is a big paper in that there are a lot of figures to it, and they actually use a lot of techniques. So we're going to talk about a number of those techniques today. Okay. Just have to figure out how to, there we go. Okay, so transfecting cells with a cDNA. You guys know how to make a cDNA, right? You insert your DNA of choice into a vector. You're going to use some kind of antibiotics to select for the cells that are expressing the DNA and, in this case, the neomycin resistant gene, right? If they grow, they're expressing both. If they don't grow, they aren't expressing. So when you use this kind of transfection, right, and there's different ways to transfect. You can use electroporation, you could use lipid, you can also just use heat shock. But the cells that get transfected are usually only the ones that are proliferating, right? The ones that are dividing. The efficiency of transfection is not actually that great. It's a pretty low percentage of cells that get transfected, and the insertion of the genome ends up being pretty random. Well, it is random, right? It's not precise. It doesn't end up exactly where you want it. Okay? So that would be for normal DNA plasmids. So you can also use retroviruses to transfect. Well, and it's actually, there's a different name for it. Okay. So that's what this paper uses as a retrovirus. So we're going to talk a little bit about viruses and what transfecting with the retrovirus means, okay, versus, you know, what it normally does. So here's a general virus life cycle. It's kind of ironic that we're talking about viruses, but you guys probably know most of this, right? Each virus infects a specific cell type, okay? Some integrate their genome into the host genome, others do not. Now, of course, if you're going to be using a virus as a transfection tool, you're going to be wanting to use a virus that actually does integrate its DNA into the host genome, right? But eventually all viruses will activate the assembly of new virus particles by using the cell that they're living in right then, right? Because they're parasites, and they usually end up killing the cell in the process. So that's not a good thing though, right? If you want to use a virus to transfect in your cDNA, you don't want to kill the cells that you want to look at. That would be bad. Okay, so what do we do? Oh, and by the way, that was a very simplified idea of, you know, how a virus works, because yeah, it's always more complicated than you think. So this is a retrovirus. So a retrovirus is an RNA virus, right? It has reverse transcriptase within that virus. Let's see. Okay. And so it enters the cell. That's because a retrovirus is that they usually can enter the cell by merging with that cell membrane. And they lose that envelope. That RNA gets out, and the reverse transcriptase will, you know, make one strand of DNA, and then it'll come back, and another strand will be made. So now you've got double-stranded DNA that can get inserted into the host genome. So if you notice in the red, right, so there's one enzyme, reverse transcriptase, and then there's another one called integrase. That's the one required to put that DNA that came from the virus into the host genome. From there, whenever, you know, that it gets activated to start making more viral particles, then we have transcription of that particular DNA, translation, and the things that it makes in order to make the particles are capsid proteins, envelope proteins, reverse transcriptase, and integrase, right? So all those things have to get contained into the viral particles. And that whole assembly also requires something called a packaging signal. Okay, hang on, I get my mouse back. Okay, so which parts of this life cycle need to be shut down for retroviruses to be useful to introduce DNA into cells? Say it louder. So if you shut down the integrase, so the point is that you want to put the DNA of choice, you know, into the cells. And I suspect I have people talking, but I can't see their chats right now. So hang on. I still see yeses.
Isn't it normally the lysis of the cell that once the newly assembled viruses are released?
Yeah, so the lysis of the cell is killing it. Okay.
So even though that, so like we have to like secrete them through the cytoplasm.
Okay, I think you're getting there.
Just stopping the virus from making more copies of itself. The transcription that leads into the translation of the proteins. Maybe if you wanted to overexpress protein, you can remove anything after the promoter and then hook up whatever gene you want to be expressed to.
Yeah, I think between the two of you, did somebody else have their hand up?
Oh, I was guessing that maybe you get rid of the packaging signal, so they won't be able to form the viruses to first open the cell.
Okay, you guys are definitely getting there. Okay, so really, you don't want any of that packaging to be going on. So that would be making the proteins, the capsid protein, envelope protein, reverse transcriptase, and even the integrase, okay? How about what needs to be kept though? Andrew? Yeah, you want to keep the reverse transcriptase, yeah. Let's see, Sebastian says the assembly of new viruses needs to be stopped. Yep. So this is what the retroviral genome looks like, okay? So you can see in the green that is some of the genes that are being expressed. So many of the components needed to actually make the virus get separated out from the actual genome. Background. Okay, so this is what the engineered viral genome looks like. So there's actually still, there's that packaging, if you go back, there's this packaging thing here. But all you really need is the cDNA and the neomycin resistant gene. Okay, and then everything else that's needed to make a virus is provided by a cell line. So the cell line actually is expressing the PAL, the integrase, and the reverse transcriptase even, okay? So you actually only need the DNA that you need and then the packaging. So that the point is that the retrovirus, or your cDNA, can get into the cell but cannot get out, right? It's stuck there. So you are transforming the cell. Yes. So it's not the cell line that you are, it's another cell line. So kind of think of this as, you want to make retroviral particles to transfect your cDNA into whatever cell line you want to use. So to make it, right, first you have to create, you have to express, you have to package basically that genome. Does that make sense? Exactly. We are not using retroviruses that have their, but they have to be packaged so that they're viral particles so that they can infect the cells that you want them expressed in. So they still need to look like viruses, but they're not going to have all the DNA inside them, the genes that express all those things that turn them viral, I guess would be, right, that are actually they're going to use to get out of the cell. So they can get in, but they can't get out. Okay. So now retroviruses are actually pretty commonly used to make cell lines that have stable expression of different genes, and lentiviruses are quite often the ones that are used the most, and the lentivirus would be HIV, by the way, is an example of one of those. So you can genetically engineer those viral genomes, right? Just like I showed you, and then generate retroviruses in cell culture. So, right, you are, like, just as I was saying, you got that cDNA that you made, it packages it up, so you now have a retrovirus with just the cDNA you want it to have, none of the other stuff to make the particles, right? So then you can infect the cells, select with the G418, and get the expression of your DNA, right? Get your protein expression. Okay. So this is what they did in this paper, and it was 24 different genes that they ended up doing this with that they thought might induce pluripotency. First, they actually had to create a knock-in mouse. They used they wanted a beta geocassette. So if you look here under A, you can see that they used a locus for a gene called FBX15. And if I'm not entirely sure why they used it. I think it was kind of ubiquitously expressed in mouse embryonic stem cells and early embryos. And it's a benign, like it's not something that's going to affect anything else. So that's what they ended up selecting to do this with. I honestly couldn't ever find a really good reason why. I did some research. Yeah. Yeah. Yes, it's J85. You haven't read it yet? Well, that's fine. You have until Wednesday. So, okay, if you look at that figure, then what they, on the bottom of it, right? You see 5' LTR, you see, so this is actually the engineered cDNA with the packaging there, and that is transfected then into these cells that they created from the mice. Okay, so they isolated both mouse embryo fibroblast cells and tail tip fibroblast cells, and then transduced both of the cells with this engineered cDNA, the 24 different candidate genes. Okay, so when you are using a retroviral vector, it's all actually called transduction, not, so. And if the transcription factors induced pluripotency, then you would have resistance to the G418, right? Because it's hooked together. Does that make sense? I should pull up my chat screen again. Okay. So I'm going to go through this pretty quick because we have a lot more to talk about, but this is kind of a refresher, right? Transcription factors. So that mediator complex, that big purple thing there, that's really, really important. And it's the thing that turns on the gene expression, okay? It's about 28 protein subunits. It includes kinases. So the basal complex, which is kind of the blue with all the different brown transcription factors attached to it, it is required, but it isn't actually turning the gene on, right? It's the mediator complex that links that activator, right? You guys you had a quick question on this, I think, along with, that whole holoenzyme, I think the whole thing is called, to turn on expression of that gene. Okay, so when we think about control of transcription, there's a lot going on. So there's multiple general transcription factors, okay, activators, repressors, mediators, co-activators, there's a lot. Then there's multiple regulatory sequences themselves. Some are near, some are far, and they bind sequence-specific transcription factors. And then, and this is kind of the point of today's lecture, we have epigenetics. So chromatin itself, right, has an impact on transcription because it's all about, is it available? Is it accessible for the transcription factors, right? Okay. Let's see. Okay. Hopefully when they record this, they record people's comments. We'll find out. Okay, so we're going to talk about measuring gene expression. There's a couple different things, and then we're going to get into epigenetics. So transcriptional profiling, looking at the end products, right? So you can use reverse transcriptase, RT-PCR, you can use microarrays, but if you use RT-PCR, what is your starting material? What's your final product? Do you need to know the DNA sequence? That's right. So that's kind of the biggest thing is, do you need to know the DNA sequence? With RT-PCR, you kind of do, right? Yeah. Okay, but the DNA microarray is a little different. So you don't, you can look at expression of, of messenger RNAs if you just look for poly A tails though, right? So you could come up with some primer that just identifies those. But overall, you're not going to profile a whole cell with RT-PCR. But you can do microarray. This is for profiling as a discovery though, not as much as, you know, coming up with a hypothesis and, you know, proving something. It's more what is out there, what is being turned on, what is being turned off. So when you look at one of these, and there is one of, they did a microarray in this paper. So the red means that the expression of that gene is upregulated, green means it's downregulated. And you can see on the top that it's showing all of the different tissues of the body, okay? So first you could do the microarray, look at what's upregulated or down and, you know, whatever you're interested in. Then you can look at, use RT-PCR to confirm this data, okay? So the next thing to measure gene expression is look at it epigenetically. So we're going to talk about two different methods, modifying histones and then looking at the methylation status of the genome. So you all should recognize this picture. I think I show this in about every class I teach, right? So you know, this is DNA packaging and it's chromatin, right? And it's the whole beads on a string form of chromatin, and it packs them into nucleosomes. And so it's DNA wrapped around, what are those proteins called? Histones, yes. Okay, and you can actually, you know, you can take a nuclease and it'll digest away the linker DNA, so you get just the DNA wrapped around the histone. You know, they did this obviously to figure out all of the structure of this, okay? In the histones, there's, you can see at the bottom, H2A, H2B, H3, H4. Okay, so those are the proteins that make up that whole histone package. Okay, so these histones can be modified through a remodeling complex, and there are different ones out there, but an example is, I think it's Swiss-Sniff. I'm not entirely sure, but we'll go with that. So this remodeling complex has 11 subunits, which include a DNA binding domain and an ATPase domain, okay? It's found in eukaryotes from yeast to humans, and the action of modifying the histones through this remodeling complex, because that's the name, right? A remodeling complex tells you what it's doing, is to make areas of the DNA more accessible to other proteins, i.e. transcription factors, right? So in yeast, the Swiss-Sniff complex causes nucleosomes to slide down the DNA strand and alter their position. And if you think about it, right, if we go back to the previous slide, it's really hard to turn on gene expression if the DNA of that gene's wound around a histone. So it makes sense that you are going to have these protein complexes that can move the histones, or kind of, you know, like it's really, you think beads on a string, moving the bead along the string, exposing the DNA sections that you want to express at the time. So the DNA can flex around that nucleosome. The subunits of the nucleosome can also be swapped out, and those, they can slide. So chromatin is actually very dynamic. It's not, you know, stagnant and just sits there, just, which is always the problem with every picture in a biology textbook. You look at it and you think that that's what's going on in your cell. Everything's just sitting there. But in reality, everything is constantly moving, right? Yeah. So the ATP hydrolysis can allow that DNA around, that's wound around the histone to loosen up and allow it to move that way. Okay, and it can even transfer histone cores from one to another. So it's pretty complex in how it works. So there's two different ways activators can turn on transcription. The first is with what we were talking about, the Swiss-Smith. So here, if you look on the top, you see that gene activator protein, right? We've talked about this enough. So it's interacting with the DNA and with the chromatin remodeling complex and some energy from ATP, the nucleosomes are now remodeled, opening up that DNA so that general transcription factors, RNA polymerase, the holoenzyme can all get in there, turn on transcription, activate transcription, right? So it's moving the nucleosomes out of the way of the promoter in an ATP dependent manner. So that one gene activator protein is right involved in this. And if you think about MyoD, right, that's that master regulatory protein. That's probably most likely, how it's involved. Another method is using histone acetylase. Okay, and you can see there's still a gene activator protein, but the histone acetylase modifies the histone protein tails on those histones. Okay, so, and then that can recruit other activators and co-activators, as you can see in this slide with the triangle shaped object. Okay, that then can activate, right, and leads to ultimately the same thing as the other method, right? Activation of transcription. Okay, so what this is telling you is there are different activators out there that can turn on transcription. And it will definitely depend on what cell type or what transcription factor or gene activator protein it is, which one ends up being used. Okay, so there's different methodologies. Should be a lot to do. So acetylation will cause a conformational change in those core histone proteins because the lysine-rich tails actually bind very tightly to the DNA and blocks access to the DNA template. And when they get, so the DNA itself is in the dark black, and then the lysine-rich tails are kind of the ones that look wrapped around. So and then the acetylation of the tails gives them, right, they're now sticking out away from the histone. Okay, so it's altering the structure and allow transcription factors to bind because acetylation is neutralizing the positive charge of the lysine. You know how, right, chemistry, how much of a difference that makes. So it's all chemistry. So the HATs, the histone acetyltransferase can do this. Also HDACs, histone deacetylases can affect this too, right? Because they can. So this kind of sums it all up. It's right. So there's two different ways, and this is there's probably more than this, but these are two major ways that have been discovered. This whole remodeling, it also can go backwards because sometimes you do want the nucleosomes to block, right, access to that DNA. So you can think of it as cyclic. You could have remodeling complex A, opening it up so that DNA binding proteins can get in, turning on gene expression. And then you can have remodeling complex B come in, cause those DNA proteins to come off and restore it back to where it was. Okay, which makes sense. Pretty much everything in the cell has an on-off switch, right? If you start thinking about it, yeah. So, okay. So this would be the pathway, right, for transcription activation. But notice the little red star at the top. It's all starting with that gene activator protein. And that's essentially what JA4 talked about, and also JA5. Okay, because they're looking to figure out how to induce pluripotency from a, cell that is differentiated. Okay. So multiple histone modifying enzymes are going to act to regulate gene expression. Okay, so you can acetylate here. So you can open it up so that you get expression, right, by deacetylate or, deacetylase, demethylase, sorry, okay, in HAT, or you can repress it with the deacetylase and the histone methyl transferase. Okay, so it's, again, on-off switch. So okay. And all these different modifications of histones form what's called a histone code. And you do not have to tell me all about this picture at all, but it just gives you an idea of, there's really a lot of different things you can do to it. In addition to acetylation and methylation, you can also phosphorylate it, and ubiquination also can play a role. So if you just look on the right side, the modification state is one row, and then the meaning of that is another. So if it's an unmodified, maybe the gene is silenced. If it's acetylated, now you could have gene expression, right? If it's phosphorylated, it might be undergoing mitosis or meiosis. Okay, if it's phosphorylated and acetylated, you could have gene expression, right? If it's methylated and phosphorylated and acetylated, well, who knows what's going on? If it's just methylated, the gene may be silenced, right? So there's a lot of combinations, which of course makes it more complex to understand, but it does make sense because the cell is constantly deciding what it needs. Does it need to turn on gene expression or not, right? So what you can do to knowing that your DNA has these modifications, phosphorylation, methylation, acetylation on the DNA, can actually use them to pull out active chromatin, okay, through immunoprecipitation. So you're going to have an antibody here, and you're going to pass a mixture of proteins through a column with a matrix that consists of beads upon which antibody is attached. So the beads are gray, the antibody are these little black things, and then any antigen that recognizes that antibody would be caught, right? And then you could rinse it and, get it to release and collect out that particular protein. Okay, and so one of the methods they use in this paper is called the chromatin immunoprecipitation method, or CHIP. So as you can see here, so it's literally looking at what proteins are binding to the DNA. So you're looking at transcription factors, possibly RNA polymerase, right? Anything that's binding to the DNA. You fix the chromatin in living cells, you sonicate it to break it apart, then you purify and immunoprecipitate, just like I showed you with the antibody, okay? And then you can use hydrolysis to reverse the cross-linking and release your transcription factors or proteins or whatever, and that piece of DNA, and then you can analyze that DNA sequence, usually by PCR, to see what the sequence was that was binding the transcription factor. So here's an actual experiment from a paper, and if you want to look the paper up, the references at the bottom. So here they looked at promoter-bound versus elongating transcription proteins. There are there's a GAL1 promoter and a GAL1 open reading frame domain. So you can see there's two different areas where they looked at binding. And what that background noise is. So if you look down at the GAL1 promoter, okay, so it's induced by the presence of galactose, and it shows it over time. And there, the promoter versus the open reading frame, right? You can see that polymerase 2 is recruited both to the promoter and the open reading frame, right? Because you see expression there, right? But the TATA TTBP is only expressed in the promoter region. It's not expressed, sorry. It's only binding to the promoter region, but not to the open reading frame. Does that make sense? Okay. This isn't actually what it, I mean, that just kind of shows you, you know, how this works. So we're next going to talk about methylation status. So you guys know all about DNA methylation, I'm sure. So you have 5-methylcytosine. The target sequence is these CPG islands, right? And methylation is associated with inactive genes, so the degree inversely correlates with the level of activity. And the methylation patterns are tissue specific. It is required for X chromosome inactivation in females. 5-azacitidine treatment turns on or up genes, right? They use that in the last paper, okay? And it actually has been used to turn on embryonic fetal beta globin genes in sickle cell patients. Let's see. So cytosine, methyl C is similar to T and U. I think this is my next slide anyway. Okay, so yeah, methylated C is similar to T or U. Okay, so this is what, if you look at this, this shows you what methylated cytosine looks like. And interestingly, the methylation status can be passed down to daughter cells. So this is epigenetics, right? This is your exposure possibly to your environment can change you. And they've done lots of studies on twins, right? As they get older, even though they look alike and have the same DNA, because of their environment, one could have cancer and one does not. Okay, so this is epigenetics for sure. So what the way that methylation can be passed on to the daughter is that there's a enzyme, of course, called methylase that recognizes these methylated CPG islands, and they then will keep the methylation, okay? Whereas if it's not methylated, it won't keep it, right? And let's see. Yeah. So it will, that methylase will continue methylating those methylated CPG islands, okay? Yeah, so it continues, gets passed on. So when there's active transcription going on, there's very little methylation on that DNA area. So the cleared areas are actually called the CPG islands. What else do I want to say about this? So they're going to be found in the promoter regions for sure. Let me get this. This is blocking my notes. And yeah, it's not going to help. So definitely housekeeping genes are going to have, you know, no methylation on them, whereas other genes that are more highly regulated are going to have some methylation going on, right? So you could have things that need to be, you know, transcribed all the time, or but being able to measure the methylation is a way to tell what if the gene is turned on or not. So it's a way to measure gene expression. Okay. actually doing okay. So here's an example of the effect of DNA methylation. So in mice, okay, IGF-2 is required for prenatal growth. But interestingly, okay, so there's 2 chromosomes, right, that have the genes for IGF-2. The paternal chromosome is the only one that's active. The maternal one has an insulator element between the gene and the enhancer that is methylated, meaning it's not going to be expressing the IGF-2 gene from the maternal chromosome. Okay? Also in mice, the IGF-2 receptor gene is also imprinted, but here the receptor is only active on the maternal chromosome. So it's the opposite, right? So the IGF-2 protein itself is expressed from the paternal chromosome. The receptor for IGF-2 is expressed from the maternal chromosome. And if you do a knockout mouse, okay, so where you don't have the paternal IGF-2 gene being expressed, you get dwarfism. If you don't have the maternal IGF-2 receptor gene expressed, you have dwarfism. If you knock out both, the mouse is normal sized. So that's pretty interesting. Just methylating that, really, that's a huge impact. So if you want to know more, there's a link in the notes section of the PowerPoint. Okay, so looking at how DNA methylation represses gene expression. So certain proteins are actually going to bind preferentially to methylated DNA, and that means that they could recruit repressors or histone deacetylases and then cause chromatin remodeling to make it actually more condensed. Instead of opening it up, it's condensing it more to completely like turn it off, okay? So, but Drosophila on the other hand doesn't methylate DNA. So it will depend on the organism. So but DNA methylation can change the gene expression pattern. So this kind of shows you how that could work. You have gene regulatory proteins binding the DNA, turning on the gene, okay? You lose the regulatory proteins, you still could have some leaky expression of the gene. If you have new DNA methylation though, and then that binds proteins that recognize that methyl C, and then more binding and activity of chromatin remodeling complex and histone deacetylases occur to completely turn the gene off, okay? So this is just another way to regulate gene expression. But you can actually measure DNA methylation status through something called bisulfite sequencing. So here you do need to know the starting sequence. Let's see. So you add bisulfate, and bisulfate will turn normal C's into U's, okay? But methylated C's stay the same. So then you can do PCR on your allele and then sequence that PCR product. Okay, so that will give you, actually it looks like this. Okay, so this is what you see. So the open circles are non-methylated CpG islands and the black circles are methylated CpG islands. So it's the same promoter, okay, different cell types, but the same promoter. So the question is, which one is expressing that gene? The right or the left? The right. What do you think? If it's non-methylated versus methylated, which of these is expressing the gene? If you methylate though, what are you doing to the gene expression? Yeah, so the promoter that's active is the one with all the open non-methylated CpG islands, right? Make sense? Yeah. Okay, good. So this is in the paper. They use this methodology. And just to show you the chemistry of it, right, how bisulfite converts cytosine to uracil, this is it. I'm not going to expect you to tell me about this. It's just kind of, you know, good to know. And quickly, the last thing is flow cytometry. We're going to talk about this in more detail for JA6, but they do a little bit of flow cytometry in this paper. Usually they're using fluorescently tagged antibodies to cell surface proteins, and they just kind of run cells through. In this case, when you start reading the paper, they use GFP to tag the cells, so that's what they're pulling out. They're counting cells by using the expression of GFP. Okay, so this is a good way to sort cells, and in JA6 we're going to get into that in more detail, so you'll learn more about that. So I know that it's like one of the longer lectures I have. Questions from the online community. You guys are all have to go. Yeah, flow cytometry is very cool. I feel like there was something else I want to say. So, look at your emails daily, because who knows, things change so fast. I will use this lovely Zoom meeting on Wednesday. I will figure out how to set up little chat rooms. So each group I'll assign you your figure. I'll tell you what you need to do, and then you can get in your chat room and talk about it. And then we have to figure out like how to share with everybody. Yeah, so I think when people talk, right, everybody will mute except the person or two in the group that's talking. And it might take a little longer. That's one thing that we have to be prepared for is, right, it will take possibly a little longer just, to coordinate everything, and, can't talk over each other because it's a lot harder to hear, so. Okay, so we're agreed that the exam, I'll post it at the end of next week, right? And you'll have all of spring break to do it. And I hate to do that. I'm against doing that on your spring break, but we kind of don't have a choice. Plus, it actually makes it better for you guys. I will work through exactly how I'm doing that. So you were saying, Dr. Miyamoto, one of you was saying that are you printing out the whole exam and just filling it in, or is it just figures? It was the whole. Okay. So did you do it already, or she's told you that? Okay. So the whole thing is on a Word document, and then you type into it, but you can print out and draw and all that, and then you're going to upload it. So none of it is actually on this. Okay. 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 Well, I will think about it. So. Okay, if you don't have any questions, I can't think of anything else. I will see you virtually on Wednesday. And please email me if anything comes up, technology problems, whatever. Okay? Thank you all. Thank you all online for attending.
Amazing.
Okay.
Everyone, take care.